Hello, my name is Jonathan Parks, and I am the Business and IHP Lead of cGEM Guelph. Today, along with my colleagues, we are going to be introducing you to our CRISPR technology and how we plan to counter climate change as a result of our project undertaking. Presenting alongside me today, we have Nathaniel Williams, our Director of Research, Danielle Halash, and Arthur Karpatov, two lab members from our research team. We also have Kristen Simule from our engineering and modeling team as well. Climate change is having a significant impact on agriculture. As average global temperatures are rising, we are seeing an increase in extreme weather conditions, such as flooding, droughts, wildfires, and more. We are also seeing warmer temperatures in the summertime and colder ones in the winter. Here we are beginning with our own Integrated Human Practice Team, also known as IHP. Our team has worked to generate insight from external stakeholders on our project undertaking, identifying CRISPR technology and its counter to climate change. We believe it is essential to diversify the feedback, and that is why we had targeted a mix of academic, industrial, and community stakeholders. The purpose of our Integrated Human Practice Team is to maximize our project's potential so that it may serve the needs of food scientists, farmers, agricultural firms, as well as communities impacted by climate change. It ensures that there's an applicability of our project undertaking to real-world problems.
The abnormalities brought on from the rising global average temperature due to climate change, such as extreme cold and drought, has had startling impacts that have placed major stressors on agricultural resources worldwide. Many of our current methods of increasing crop production are not sustainable, such as the use of pesticides and fertilizers. At the current rate that the environment is changing, many plant species are struggling to develop the mechanisms needed to withstand climate change by natural means.
In broad strokes, we intend to use CRISPR activation technology in order to increase the expression of specific genes involved in plants' natural response to environmental stressors. The expression of the CRISPR activation system itself will in turn be controlled by an inducible promoter, which should allow us to temporarily initiate the overexpression of the target genes by exposing the plant to an inducer molecule. We begin our research with two primary hypotheses which will be tested over the course of the project. First, that overexpressing our chosen target genes while the plants are exposed to the corresponding environmental stresses will assist the survival and health of the plants. These genes are ICE2 to assist with cold tolerance and CLE18 to help the plant survive in drought conditions. Second, that the use of an inducible promoter will be sufficient to control the expression of the modified CRISPR system, so that the overexpression of the target genes will only occur in the presence of the inducer molecule, either ethanol with the alcohol promoter or beta-estradiol with the XVE promoter. My colleagues will now explain each part of the experimental system in greater detail.
The plant Arabidopsis thaliana will be used as the experimental model for our genes of interest. It is a fast-growing and well-studied specimen with a fully sequenced genome that shares genes with many other staple crop plants. Knowing this, the research completed in this project has the ability to be translatable into other agricultural species and will hopefully set a positive example of the use of synthetic biology as a solution for environmental issues, prompting more research. There are 4 target genes that will be over and underexpressed using the CRISPR systems. In order to specifically increase drought and cold tolerance, we are interested in the 2 genes, ICE2 and CLE18. ICE2 within our model plant species has the effect of increasing cold tolerance when overexpressed. This allows for better growth into cold temperatures within the range of approximately -5 to -20 degrees Celsius. The overexpression of ICE2 also has been found to decrease the amount of damage caused to rosette leaves in cold temperatures. The actual purpose of ICE2 is to code for BH-LH factors, which regulate the expression of many cold-responsive genes. The second gene of interest, CLE18, is a root growth factor. When overexpressed, it results in long and wavy roots, partially by regulating the root apical meristem within plants. The aim of overexpressing CLE18 is to improve drought tolerance within A. thaliana by enhancing root growth length and waviness. DXR and SPCH will be used as diagnostic genes to indicate if CRISPR-A and CRISPR-I are performing as expected. DXR is responsible for leaf color, an easily observable phenotype. DXR overexpression results in high pigmentation and underexpression in low pigmentation. SPCH is responsible for stomatal development, and underexpression of SPCH reduces the amount of stomata present. This is very useful to us because ICE2 is a transcription factor for SPCH, and therefore SPCH will most likely have lower expression levels when ICE2 is overexpressed, compared to SPCH alone targeted for overexpression.
Clustered regularly interspaced short palindromic repeats, or CRISPR, is the most exciting genome editing technique currently used in synthetic biology laboratories. It is derived from the native immune system found in bacteria used to defend against viruses. CRISPR can be used to inactivate a gene or replace a gene, and a modified version of CRISPR, CRISPR activation, or CRISPR-A, and CRISPR inhibition, or CRISPR-I, can turn genes on and off, respectively, as a molecular switch responding to changes in the environment or the introduction of specific chemicals into the nucleus. Two components of CRISPR allow it to be effectively used in genome editing: a guide RNA with complementary nucleotides to that of the target site in the genome, and a nuclease enzyme called Cas9, which effectively cuts the DNA when the guide RNA of CRISPR binds to the target site. We will be utilizing CRISPR-A to upregulate and CRISPR-I to repress our genes of interest. This technique uses a mutated version of Cas9, dCas9, which lacks exonuclease activity and rather serves to deliver the transcriptional regulator. CRISPR-A can increase or decrease the rate of transcription according to our guide DNA, and the transcriptional regulator that we'll be using for activation is the transactivation domain VP64 bound to the C-terminus of the dCas9 domain. This is a tetrameric repeat of the herpes simplex virus minimal activation domain VP16. Meanwhile, CRISPR-I represses transcription. The transcriptional regulator for repression of genes of interest will be the CRAB KRAB repressor domain, and this CRAB repressor domain comes from the human zinc finger protein ZNF10.
Let's take a closer look at the function of guide RNAs within our project and how we plan to maximize their efficiency. Within the promoter region of the target genes, there exist many different PAM sequences which can be recognized by dCas9, with the efficiency of each location varying in their ability to allow the dCas9 complex to recruit or block RNA polymerase. For each of the 4 genes that the iGEM Guelph 2020 team will be targeting with the CRISPR system, 5 scaffold guide RNAs, or sgRNAs, will be designed, meaning that 20 different guide RNAs will be involved in the project. Each of the designed sgRNAs will be tested to see which region within the promoter of their respective gene of interest is most optimal for the CRISPR RNA portion of the sgRNA to bind. The guide sequence that results in the strongest phenotypic change in each gene of interest will have its CRISPR RNA used in all further experiments that involve the target genes and CRISPR. A further process for testing sgRNA efficiency is a process known as multiplexing. Multiplexing as a tool in CRISPR systems involves the use of multiple sgRNAs at once to increase the expression of target genes. This method of testing for sgRNA efficiency was proposed in the research completed in 2013 by Albert Cheng and others. This process will be utilized in the CRISPR implants project when working with dCas9 with the VP64 transactivation domain. The results of both the testing of individual sgRNAs and combination of sgRNAs will be compared, and the method that results in the strongest phenotypic expression will be utilized in the project. The iGEM Guelph 2020 team will be utilizing a relatively new form of molecular cloning technology known as Golden Gate cloning. Golden Gate cloning functions by using type 2S restriction enzymes to cut outside of recognition sequences found within DNA to result in scarless cloning. This provides an advantage over traditional cloning methods that commonly have issues with DNA strands religating at the cut sites. One example of a type 2S restriction enzyme is BSA1, which will be used in this project. The plasmids that we're using are equipped with 2 BSA1 restriction sites encoded within the DNA that when cut produce non-complementary overhangs, ensuring that an entire section of the plasmid will be removed when both sites are cleaved. This ensures that if the old sequence religates, it can immediately be digested out again until the target sequence is inserted. The inserted sequence does not contain BSA1 recognition sites and thus will not be digested following insertion. Golden Gate cloning will be used in this project as a tool to insert the designed sgRNA sequence into our plasmid backbones. The sgRNAs are designed with overhangs that match the cut sites created by the BSA1 enzyme within the plasmid. The large quantity of sgRNA that we will be testing requires means of efficiently swapping their positions within different regions of the promoter, and thus Golden Gate cloning would be the most optimal tool for this project.
The use of inducible promoters comprises the key novelty of our project. We don't want the target genes to be expressed all the time. For example, there are some drawbacks when overexpressing IS2. This involves the delayed flowering of the plant and reduced growth in seedlings. Our aim is to essentially create a human controllable response in plants. And the inducible promoters that we've been looking into are the ethanol gene switch designed by Caddick et al. And the XVE beta estradiol inducible promoter designed by Zhu et al. So the testing of the genes will be done by reporter plasmid with green fluorescent protein. And again, the inducible promoter will control the CRISPR system, making the system humanly controllable. Now the diagram on the slide here illustrates that the activator protein only binds the inducible promoter when the inducer molecule associates with the former, thus causing the downstream gene to be expressed.
One potential obstacle that we may face is if the gRNA sequences we have selected do not correspond to optimal target sites in the promoter regions of the selected genes of interest. This could be the case if the positioning of the CRISPR-A Cas9 complex were to block the formation of the transcription pre-initiation complex, thus defeating the purpose of CRISPR activation. If this were the case, no increase in mRNA levels would be observed. One solution to this problem would be to redesign the gRNA sequences to target different regions of the target gene promoter. However, since we are already testing multiple guide sequences for each target gene, it is unlikely that none of them will work. Given that Arabidopsis is a natural response to both ethanol and estradiol, there is also a real possibility that the inducer molecules we are using will activate unwanted response pathways in the host, potentially interfering with the effectiveness of the CRISPR-A and CRISPR-I systems. For this reason, we will test each candidate inducible promoter using the beta-glucuronidase gene as a reporter, and we will also test the effects of ethanol and estradiol on wild-type Arabidopsis. Should both inducers prove detrimental to our goals, we will attempt to use other methods, such as injecting liposomes containing the gRNAs into the host, as has been done in 2018 by iGEM BOKU Vienna.
The desired end results of the project are to provide a proof of concept for our system in Arabidopsis thaliana with the long-term goal of implementing it in other plant species. We are utilizing advanced technologies such as Golden Gate cloning and follow streamlined protocols to ensure an unmodified genetic sequence, but with the changes in protein expression we want. That is, the portion of the plasmid containing our CRISPR system will be inserted randomly into the plant genome by recombination, so the plant's genome will be modified, but the target gene sequences will remain unmodified. This will allow for the controllable expression of genes that counteract climate change and can be applied to valuable crops. For example, in the event of a late frost, we could induce the overexpression of ICE2 for the duration of the cold snap. If successful, the findings are believed to be applicable to crops threatened by climate change that have greater value. Thus far, we've made the genetically modified plasmids, transformed the agrobacterium that will be put into the Arabidopsis thaliana, and our next step is to actually introduce it into the plant.
The engineering and modeling team have been focused on representing the expected results of genotypic and phenotypic changes for our mutant Arabidopsis plant. As this is the inaugural year of iGEM Guelph's engineering division, we have been guided by Braden Kell, a former dry lab lead for iGEM Toronto, co-program director of CSberg, and current PhD student in biophysics. Braden was able to direct the modeling team by providing informative video guides, accessing literature, and contacting colleagues for added assistance. As previously mentioned, we are interested in altering the expression of certain genes related to climate change response. Time-dependent modeling techniques will allow us to account for the lag time between promoter induction and increased rate of transcription. We will tag individual sections of mRNA to continuously collect data for the entire transcription process. By using the mathematically determined elongation rate and known length of mRNA, we will be able to find both lag time and time of noticeable increase in gene expression. Following these molecular changes is a noticeable change in the plant's phenotype. Qualitative changes are tracked using colorimetry and root elongation software in replacement of human error-fueled physical observation. These same methods will also be used to quantify DXR expression to determine the plasmid's effect on the DXR gene controlling chlorophyll pigment and coloration in the plant. The promoters mentioned will be induced by ethanol and estradiol. Ethanol stunts the uptake of water in plants, while estradiol enhances root growth and biomass. Both molecules produce these effects by activating the inducible promoters, therefore inducing the expression of our CRISPR system, which regulates transcription of the target genes. We will analyze the difference between wild-type growth and treated mutant plant growth. With regards to engineering, it has been challenging to develop projects during the COVID-19 pandemic. Regardless, our team has learned to effectively use Fusion 360, a 3D modeling software, to develop our own model of an incubating shaker. This software was beneficial to our team because it is a free, user-friendly program that works online with easily shareable components. Because our team worked remotely, the software made collaboration as efficient as possible. Once construction is complete, we will perform a materials efficiency and comparative analysis of our shaker to ensure that it is cost-effective, durable, and up to industry standard. Working with both the research and modeling team, we will begin testing the shaker's quality and ability to replace the on-market model. In order to accomplish this, we will conduct a temperature test to make sure our shaker stays within the optimal range for bacterial growth. We will also perform a rotation test to ensure that the motor and shaking apparatus provide the appropriate rotational frequency. Additionally, a growth model was developed which we will use to guarantee that the overall environmental growth needs of the bacteria are met. Through collaboration with the research team, our team aims to provide engineering solutions and future ideas to implement the research. Once the design is finalized, we hope other teams will be able to take advantage of the technology to develop their own incubating shaker to be used in biological research and bacteria growth. Integrating Braden's advice on using a growth assay to develop continuous data to assess the proliferation of the bacteria, we hope to model how well agrobacterium transforms. To model transformation efficiency, the modeling team will collaborate with the research team to conduct a residurin assay. This experiment will provide us with a steady stream of data to effectively analyze the population of the specimen changes following modifications.
As previously identified, we want to ensure that our project undertaking applies to stakeholders and offers solutions to their problems. We are confident in the potential our project has in transitioning into a functional business. Here we would like to identify the entrepreneurial potential aligned with our CRISPR technology and its counter to climate change. Working alongside integrated human practice, developing an understanding of the market contribution our project has, we are working to develop a strong business framework involving marketing, financing, and operational strategy to develop a competent model which we may execute in industry upon completion of our project at a high level. As part of our project undertaking, we wanted to take the opportunity to engage within the community and offer insight on our project undertaking, CRISPR technology, and how we plan to counter climate change. We had the opportunity to connect with the Guelph Wellington Men's Club and present to them about our project. The Guelph Wellington Men's Club is a group of educated and interested driven gentlemen who wanted to learn more about our project, as well as offered insight on how we could further grow into a corporation, as well as maximize our community engagement. We also worked on our social media platforms to ensure that we were offering educational material to our followers and keep them up to date with the progress that we have achieved over the course of our project. cGEM Guelph embraces the opportunity to collaborate. With regards to our intra-team collaboration, we conducted weekly meetings representing each of our 3 sub-teams: engineering, math, and integrated human practices. We also wrote 6 literary reviews with an emphasis on synthetic biology, which is currently in the process of being published. Another example of intra-team collaboration is our engineering and research team working together to design and create an incubator in which we utilize in our project undertaking. With regards to inter-team collaboration, we participated in events such as cGEM, the Waterloo Mixer, microbes, and food production. The ability to engage with other universities was very promotive in our project. And lastly, regarding extra team collaboration, we developed a strong relationship with the Wang Lab academic group, who provided extensive training, materials, and advice for our project. As we conclude our presentation, I would like to say thank you on behalf of iGEM Guelph. We are confident in our research project and believe that Guelph iGEM will be able to aid in the counter of climate change and make a positive impact on our world. Thank you for taking this opportunity to listen to our project undertaking, and we welcome any feedback that you may have. As well, any questions we are happy to answer.